Challenges and Limitations in Brain Organoid Research. Chapter 1. Technical and Ethical Challenges. Section 1. Technical Hurdles. 1. Vascularization. Current status. One of the major technical challenges in brain organoid research is the lack of a functional vascular system. In vivo, the brain is richly supplied with blood vessels, which provide essential nutrients and oxygen while removing waste products. Implications: the absence of such a system in brain organoids leads to limited growth and maturation, as the inner cells often die due to lack of nutrients and oxygen. Potential solutions: researchers are exploring several strategies to address this issue, such as incorporating endothelial cells to form rudimentary blood vessels or co-culturing organoids with engineered blood vessel networks. 3D bioprinting. This technique involves printing cells in layers to create complex tissue structures, including blood vessels. Using bioinks that contain endothelial cells, researchers can print vascular networks within the organoids. Microfluidic systems. These systems can mimic blood flow by perfusing organoids with nutrient-rich fluids, improving nutrient and oxygen delivery. This technique involves creating channels within or around the organoids that simulate the function of blood vessels. Growth factors and signaling molecules. Adding specific growth factors and signaling molecules to the culture medium can promote the development of vascular structures within organoids. These molecules can guide the differentiation of stem cells into endothelial cells, which can then form blood vessels. 2. Incomplete maturation. Current status: brain organoids generally represent early developmental stages and often fail to fully mature, limiting their utility in modeling adult brain functions and diseases. Implications: this immature state impacts the organoid's ability to accurately recapitulate adult brain physiology and pathology, making it challenging to study late-onset neurodegenerative diseases. Potential solutions: efforts to enhance maturation include prolonged culture periods, optimized differentiation protocols, and environmental cues that mimic the in vivo conditions of the brain. Prolonged culture periods, extending the time that organoids are kept in culture, can allow for more extended developmental processes, leading to greater maturation. This can be supplemented with periodic medium changes to ensure a constant supply of nutrients. Biochemical cues: adding specific hormones, growth factors, and other biochemical signals can mimic the brain's developmental environment. These cues can guide the differentiation of stem cells and the maturation of neural cells. Environmental factors: mimicking the brain's physical environment by using biomimetic scaffolds that replicate the extracellular matrix can enhance cell differentiation and maturation. These scaffolds provide structural support and biochemical signals that promote the development of mature brain tissue. Electrical stimulation: applying electrical impulses can promote neural activity and synaptic development, encouraging maturation of the neural networks within the organoids. 3. Reproducibility and standardization. Current status: the generation of brain organoids is a complex process that can result in significant variability between batches and laboratories, affecting the reproducibility of results. Implications: this variability poses a challenge for comparative studies and large-scale drug screening, as inconsistent organoid quality can lead to unreliable data. Potential solutions: standardizing protocols and using automated systems for organoid production can help reduce variability and improve reproducibility. Standardized protocols: developing and sharing detailed protocols for every step of organoid production, from stem cell culture to differentiation and maturation, can help ensure consistency across different laboratories. Automated systems: implementing robotic systems for organoid production can reduce human error and variability. These systems can handle tasks such as cell seeding, media changes, and monitoring growth conditions, ensuring precise and reproducible results. Quality control measures: establishing rigorous quality control measures, such as using imaging and molecular analysis to assess the structural and functional attributes of organoids before experiments, can ensure that only high-quality organoids are used. High-throughput screening: using high-throughput screening methods can allow for the rapid and consistent production of organoids, facilitating large-scale studies and drug screening efforts. Section 2. Ethical considerations. 1. consciousness and sensory perception, concerns as brain organoids become more complex. Ethical concerns arise regarding the potential for these structures to develop forms of consciousness or sensory perception, while current organoids lack the complexity required for such experiences. The rapid pace of advancements necessitates careful ethical scrutiny. Regulatory perspectives, ethical guidelines and regulations need to be developed to address these concerns, ensuring that research is conducted responsibly. Defining boundaries, establishing clear ethical boundaries for the complexity and functionality that brain organoids can achieve. This includes setting limits on the level of neural activity and connectivity that is ethically permissible. Ethical review boards, forming multidisciplinary ethical review boards that include bioethicists, neuroscientists, and legal experts to oversee brain organoid research. These boards can evaluate the potential ethical implications of proposed studies and provide guidance on best practices. Public engagement, engaging with the public to discuss the ethical considerations of brain organoid research. This can help build public trust and ensure that the research aligns with societal values and expectations. 2. Use of human stem cells. Concerns the derivation of brain organoids from human pluripotent stem cells, particularly embryonic stem cells, raises ethical issues related to the source of these cells and the potential for creating life-like structures. Regulatory perspectives, ethical sourcing of stem cells and adherence to strict consent protocols are essential. Ethical sourcing, ensuring that stem cells are sourced ethically, with full informed consent from donors. This includes using iPSCs as an alternative to embryonic stem cells to avoid the ethical issues associated with the latter. Consent protocols, implementing strict protocols for obtaining consent from stem cell donors, ensuring that they are fully informed about the potential uses of their cells and the research implications. Guidelines for use and disposal. Developing clear guidelines for the use and disposal of brain organoids. This includes ethical considerations for the lifespan of organoids in culture and the appropriate methods for their disposal to respect the human material from which they were derived. Chapter 2. Limitations in mimicking brain complexity. Structural simplifications. Current status. Despite significant advancements, brain organoids remain simplified versions of the human brain. They lack the full range of cellular diversity, precise organization, and complex circuitry present in vivo. Implications. This structural simplification limits the organoid's ability to model certain brain functions and diseases accurately, particularly those involving complex intercellular interactions and specialized brain regions. Potential solutions: enhancing the cellular diversity and structural complexity of organoids through advanced differentiation protocols and the incorporation of additional cell types, such as glial cells and vascular elements, can improve their fidelity. Advanced differentiation protocols, using more sophisticated differentiation protocols that guide stem cells to develop into a broader range of neural cell types, including neurons, astrocytes, oligodendrocytes, and microglia. 3D bioprinting, employing 3D bioprinting to create organoids with precise spatial organization, mimicking the layered structure and cellular architecture of the brain. This can enhance the interactions between different cell types and improve functionality. Co-culture systems, developing co-culture systems where brain organoids are grown alongside other cell types, such as endothelial cells and immune cells, to better replicate the brain's microenvironment and enhance structural complexity. 2. Functional limitations. Current status. The functional properties of brain organoids, such as synaptic connectivity and electrophysiological activity, are still relatively rudimentary compared to the human brain. Implications. Limited functional maturation impacts the utility of organoids in studying dynamic brain processes, such as neural network activity, synaptic plasticity, and behavioral responses. Potential solutions: integrating advanced bioengineering techniques, such as microfluidics and electrical stimulation, can enhance the functional properties of brain organoids. Microfluidic devices, utilizing microfluidic devices to create dynamic culture conditions, such as fluid flow and mechanical forces, that mimic the brain's microenvironment. These devices can enhance nutrient delivery and waste removal, promoting better neural activity and maturation. Electrical stimulation. Applying electrical stimulation to organoids can enhance synaptic connectivity and neural network activity. This can be achieved using electrodes or optogenetics to stimulate specific regions of the organoids. Organoid on a chip. Developing organoid on a chip systems that integrate multiple brain regions or connect brain organoids with peripheral tissues, such as muscles or sensory organs, to study more complex neural circuits and functional responses. Chapter 3. Future Perspectives and Potential Solutions. 1. Enhanced vascularization. Researchers are developing techniques to incorporate functional vascular networks into brain organoids, which could significantly improve their growth, maturation, and longevity. Bioengineering. Synthetic vessels. Using bioengineering approaches to create synthetic blood vessels that can be integrated into organoids. These synthetic vessels can provide a scaffold for endothelial cells to form functional vascular networks. Endothelial cell integration. Incorporating endothelial cells into the organoid culture to promote the formation of natural vascular structures. This can be achieved through co-culture techniques, or by genetically modifying stem cells to express endothelial markers. 2. Improved maturation protocols. Advances in differentiation protocols and culture conditions aim to extend the maturation of brain organoids, enabling them to more closely resemble adult brain tissue. Prolonged culture and environmental cues. Extending the culture period and providing environmental cues, such as mechanical forces and biochemical gradients, can promote the maturation of organoids. This includes mimicking the extracellular matrix and using growth factors that support neural development. Supporting cell types. Incorporating supporting cell types, such as astrocytes and oligodendrocytes, which play crucial roles in the maturation and functionality of neural networks. These cells can provide essential support for neuronal growth and synaptic activity. 3. Standardization and automation. To address the reproducibility issue, standardizing organoid production protocols and implementing automated systems for organoid generation are crucial. Automated production systems. Using automated systems to handle the production process, from stem cell culture to organoid differentiation, can ensure consistent quality and reduce human error. These systems can also monitor the growth conditions and make adjustments as needed. High-throughput screening. Implementing high-throughput screening methods to rapidly produce and test large numbers of organoids. This can facilitate drug discovery and toxicology studies by providing a consistent and scalable platform for testing. 4. Ethical guidelines and oversight. As brain organoid research progresses, the development of comprehensive ethical guidelines and regulatory oversight is essential. International consensus. Establishing international consensus on ethical standards for brain organoid research. This can involve collaborations between regulatory bodies, scientific organizations, and bioethicists to create globally accepted guidelines. Ongoing ethical review. Continuously reviewing and updating ethical guidelines as the field evolves. This can ensure that new developments and potential ethical concerns are promptly addressed. Maintaining responsible and ethical research practices. Conclusion. The field of brain organoid research is rapidly evolving, offering significant potential to advance our understanding of human brain development, disease mechanisms, and therapeutic strategies. However, addressing the technical, ethical, and functional challenges is crucial for realizing the full potential of these models. Ongoing advancements in bioengineering, stem cell technology, and ethical oversight will be key to overcoming these hurdles and ensuring that brain organoids become a robust and reliable tool in neuroscience, research, and personalized medicine. Developing interdisciplinary approaches that combine insights from biology, engineering, and ethics will be essential for the responsible and effective advancement of brain organoid research.